Adaptimmune is a T-cell therapy company, both autologous and allogeneic, and it's going to be my pleasure to talk you through where Adaptimmune is at today. I'm Chief Business Officer. I'm also co-founder of the company, so we're nearly 14 years into this journey. And Adaptimmune is focused on T-cell receptor, optimized affinity T-cell receptors as the basis for producing therapeutic products for cancer. So we're listed on NASDAQ, so the usual disclaimer for forward-looking statements. And then just to spend a moment on why T-cell receptors. There's an awful lot of knowledge in the space in cancer cell therapy for CAR constructs, chimeric antibody receptors, which are antibodies with signaling machinery to target cell surface proteins. Whereas T-cell receptors is really what the body created to be able to access peptide fragments processed and presented from intracellular proteins on the cell surface in the context of tissue-specific molecules, peptide HLA complexes. And the reason that's important is that the vast majority of cancer targets in solid tumors, in cancers, are actually based on mechanisms of breakdown inside the cell. So accessing intracellular targets is extremely important for solid tumors, which is where we've been based with Adaptimmune from the very beginning. So today, Adaptimmune has built up an integrated capability of cell therapy research, preclinical safety testing, translational capabilities, as well as clinical in-house CMC, both making lentiviral vector and cells, the regulatory organization to support all that, as well as our initial commercial infrastructure. We're also now 2 years into a 5-year strategy we set out in 2025 to bring products to market. So we've been focused on the science for a long time in the early days, but now we're absolutely laser focused on how to bring products forward. And primarily this is focused around our MAGE-A4 targeted T-cell receptor franchise. MAGE-A4 is a cancer testis antigen. People don't really know what it does in cancer, but essentially it's on multiple solid tumors, uniquely on those tumor cells and not on normal tissues, which makes it a really attractive target for solid tumors for active T cells. So with our 2-2-5-2 strategy, we're aiming for 2 product launches, 2 additional BLAs to be filed, 5 further products to come into the clinic from our autologous preclinical pipeline, as well as 2 allogeneic products from our iPSC-derived T-cell platform. And I'm going to touch a little bit on all of those. So here is the autologous pipeline chart, just showing really what we've been doing, where we've come from, and what we're doing next with the MAGE-A4 targeted T-cell receptors. The first product, our optimized TCR targeted to MAGE-A4, the TCR alone, we took into a phase 1 study initially in multiple solid tumors and saw responses across multiple solid tumor types, spectacularly so in synovial sarcoma and myxoid round cell liposarcoma patients, and that enabled us to do 2 things. We launched a spearhead, we call our spearhead 1 trial, which is a registration-directed study for myxoid round cell liposarcoma and synovial sarcoma, which has now completed enrollment of its first cohort 1, which will be the subject of a BLA filing this year, and I'll show you a little of that data. We also then realized that whilst we were seeing responses, we needed more powerful responses and increased depth and durability with that product. And so we pivoted very quickly to a next-gen version of the therapy, which included a CD8 alpha co-receptor, which effectively powered up the CD4 compartment of the T-cell product we were manufacturing. And that forms the basis of our SURPASS franchise of studies. So with that first cohort of patients in the first phase 1 study, we actually saw responses across multiple solid tumor types that were both reasonably durable and not just indications of activity. And again, I'm going to show you a little bit of that data. It was strong enough in our minds to form the basis of a phase 2 potentially registration-directed study, SURPASS-2, in esophageal and gastric junction cancers. And we've also seen enough data to support the planning of a phase 2 study in ovarian cancer as well, which is planned to initiate this year. We had a program targeting an alpha-fetal protein as well running, which has completed phase 1 safely and with signs of efficacy, but we've decided that we need a slightly more potent TCR for that, and so we've taken it back into the preclinical pipeline to focus on commercializing with the MAGE-A4 program. So here is the data from the first cohort of the Spearhead 1 trial, a really nice waterfall plot where you see slight difference between independent review and investigator review of the same cohort of patients. But essentially, really excited to see that the overall response rate was 34% by the independent review, which is really what matters from the registration perspective. And in amongst that, synovial sarcoma had a 36% response rate, and myxoid round cell liposarcoma with smaller numbers of patients, a 25% response rate, but an overall disease control rate of 85%. So that will form the basis of a BLA filing, which is due to go in later this year. Really excited about that. We've primarily completed most of the regulatory dialogue around that first cohort, which has met its primary endpoint, will form the basis of the BLA filing and the clinical package. And now we're focused, not surprisingly, on the manufacturing elements. The release of the GMP facility that we'll be using, which in this case will be with our partner Materni Biotech. And with that, we are characterizing both the vector and the T-cell product processes, which will obviously form the basis of characterization assays, etc., that we'll need for our CMC dossier. We're also having to register a companion diagnostic for the MAGE-A4 protein as well, so that's been moving along in parallel for some time, and will form the basis of an initial submission for the PMA approval for that central diagnostic process with the BLA. So switching to the second-gen program, this just highlights data presented at ESMO last year. So data cut was actually last August, so it's a little old now, and the study is continuing to enroll. But you see two things here, the waterfall plot and the spider plot, where you see elements of the durability of the response in these patients, and including a complete response in an ovarian cancer patient there, which was actually durable past 25 weeks at the time of presentation of this data. So that is continuing to enroll, as I mentioned, in 5 focused indications, including head and neck cancer, ovarian cancer, gastroesophageal, synovial sarcoma, and lung cancer. So very excited about that, the scope of that program to yield further phase 2 studies. And we'll be updating on that again at ESMO this year. And I think also equally exciting is the safety profile of the SPEAR T cells in solid tumors across multiple solid tumor patients. We've seen an acceptable safety efficacy benefit. We have seen adverse events that have been associated with CAR-T programs, but basically at a lower level and a lower frequency, and those include cytokine release syndrome, a specific event associated with neurotoxicity called ICANS, as well as prolonged cytoneutropenia, which is something which unfortunately is consistent with the lymphodepleting chemotherapy that patients are given before they receive the cells. And all of these are manageable and have been managed effectively by various approaches, with either moderating the patients, moderating the lymphodepleting chemotherapy they receive, or indeed being a little bit more selective about the patients that we can enroll. These are very late-stage patients. So just switching to where do we get 5 more INDs coming through by 2025. We have a number of other second-gen programs progressing through late-stage preclinical development, and these include various other mechanisms to support the potency, the trafficking, and the ability of the T cells, once they're in the tumor cells, to stimulate a broader immune response. And a number of those are in-house per the top line. A number of them are via partnerships. We have one in partnership with Tayton-Martin Biotech, which is due to be filing for an IND this year, and other checkpoint modalities with Alpine Immune Sciences. So a sort of array of other second-gen approaches, which we think will be very important in terms of getting that depth and durability overall of these types of products for durable benefit in patients. We've also got other HLA types coming through. I think it's very important to recognize that if you have to screen for the cancer antigen and screen for the tissue type, that's already restricting the patient population. And with our HLA-A2 directed program, which is the one currently in the clinic, today we know that we can reach around 40,000 MAGE-A4 HLA-A2 positive patients. And if we increase the HLA restriction to include A1 and A24, then we get up to near 85%. So we obviously increase that population quite considerably. And so those are coming along fast behind. We have other other TCRs coming through and other targets, a partnership on PRAME with GSK. And then the two bottom rows on here are two other platform technologies that Adaptimmune has been looking at. The bottom one, HLA-independent TCRs, are basically TCRs that can recognize cell surface proteins specifically, and that obviously moves away from HLA restriction, which is something that we're also keen to do, and have got two target programs moving forward, one looking at GPC3, which is hepatocellular cancer targeted target, and another one on mesothelin, which is in partnership with Astellas, which I'll come on to. The other platform technology is we're also using some of our next-gen capabilities to look at next-gen TIL approaches, and we're doing that in collaboration with CCIT in Denmark, which is a well-known TIL therapy specialist center, and that CTA has been submitted, so that study will open this year. And finally, I wanted to switch to our allogeneic platform. So this is where we touch a little bit on the regenerative medicine dimension with iPSCs. We've developed over more than 5 years now an in-house capability to gene edit iPSC-derived stem cells and differentiate those into effective T cells. We actually are now able to generate CD8 single positive T cells. And we're now in the process of scaling up that process. We're building our own GMP facility, which is going to be co-located with a research facility based in Oxford, south of Oxford in the UK, with the aim to have our first product in clinical trials by the end of next year, end of 2023. And I just wanted to spend a moment on if you like, the beauty of being both an autologous and an allogeneic T-cell therapy company. And I think really the core of this for us is that we have a 6-stage allogeneic process that is highlighted along the top here, which shows you how we move from an iPSC cell to a hemopoietic progenitors through the first 3 stages of the process. And then we move further down the differentiation pathway to double positive CD4, CD8 T cells, and ultimately a single positive CD8 T cell. Where the beauty comes in is that we have an autologous product that we know is functional and effective in patients today, and is going to be the basis of registered products in the very near future. And this plot on the right is a single cell analysis chart, which I think is an incredibly powerful technology. You'll see the autologous product, a mass of 4 different cell types there. That represents the autologous product that we have in the clinic today. The red and the blue are MAGE-A4 transduced CD8 cells in the red and CD4 cells in the blue, and the non-transduced are the black and the brown. The other blobs on this chart represent the iPSC cells we start with, and they have a very different profile in that top left-hand corner. By the time we get to stage 3, the bottom pink cells, that blob is moved quite considerably. But by the time we get to stage 5, the green cells, we're beginning to get closer to the profile of the autologous T-cell product, the product we have in the clinic today. And by the time we get to stage 6, it's actually touching. So we're very excited about the Allo program. We have a number of our own programs moving forward, as well as partnerships with Astellas and also with Genentech, signed last year. Most of these are focused on specific targets, but the second half of the Genentech program is looking at personalized T-cell therapies, where any patient's TCRs can be isolated and dropped into an off-the-shelf T-cell. That's the vision, a lot of biology that we're working through with them on that, but very exciting. So finally, Adaptimmune today is transatlantic, very much so, based in Philadelphia, where our corporate and clinical and manufacturing base is, Milton Park, where the research base is, the other corporate functions, as well as Stevenage in the UK and the Cell Therapy Catapult Centre, where we have vector manufacturing, and we're now over 500 folks. Fortunately for us, we're also funded out until early 2024, given the current climate. So thanks very much for your attention. I'm around through tomorrow for any questions.